MKU since 2020. She has received Raman Postdoctoral Fellow at UCSF USA in the year 2016. She is the first rank holder right from BSc to MSc as well as MPhil. She is also the recipient of Innovative Teaching Award Gold Medalist from MKU in the year 2009. With all these credentials, we are happy to have you today, ma'am. So welcome you, ma'am, once again. I also extend a warm welcome to our principal, Dr. Sister A.S.J. Lucia Rose, our secretary, Reverend Sister Flora Mary, and our deputy principal, Dr. Sister Kulanday Teres. I accord a cordial welcome to all the heads of various departments, deans, members of the organizing committee, and finally, a warm welcome to the student participants from our college and from colleges all over India. Welcome you all once again.
Thank you, ma'am, for your warm words of welcome. Before moving into the session, let us have photo session now. Dear participants, kindly switch on your camera for a group photo. Dear participants, kindly switch on your camera. Thank you, participants. I feel pleasure and honor to invite the resource person of this session, Dr. M. Jayalakshmi, Head of the Department of Immunology, School of Biological Sciences, Madurai Kamaraj University, Madurai, to share her expertise with us. Over to you, ma'am.
Thank you, ma'am, for your nice introduction as well as invitation. Is it audible? Is it audible, ma'am?
You're audible.
Audible, ma'am. Audible. Audible, ma'am. Audible.
Thank you. Thank you. Yeah, let me share my screen. Hello. Okay. Meeting sure. Thank you. Yeah, is my slide visible? Is
yes, ma'am.
Okay,
yes,
thank you. And
yes,
yeah, thank you for your invitation once again. And I have great respect to your college. I have been visited a few times, but I was really attracted towards the discipline and the nature your college was operating. Even though in this lockdown period, I feel I am connected with you all through this online meeting, and I thank God for this opportunity. And yeah, I could see the faces of few students, and those few faces actually make me to think about my college days, and I feel I want to do justice. I will convey at least a small message to those students who are eagerly waiting to know a few basic things from us. Okay, so like as it was introduced to all of you, yeah, we are doing the immunogenetics, mostly the immunogenetic research. What makes the people different to being differential susceptibility as well as showing resistance to different kinds of diseases. So we are well aware now. Everybody is aware, and mostly this has been the topic of immunology has been brought into attraction to the whole world by the due to COVID. Everybody understands the situation, and it is clear that there is a strong host immunogenetics that is, and as well as the host immune genes are responsible for keeping few of us protected from some diseases, and few of them being affected from diseases. And even though few of them are being affected by the diseases, they can successfully overcome those diseases, and that is all because of this immune system what we are having. And that immune system is also modified by the way of the mutations and SNPs, what is actually existing in our immune response genes. So for studying all these things, we need certain basic principle methods, and the first method may be the DNA extraction from the whole blood. So I'm going to share with you today the method. One of the method what we are going to discuss or we are doing right now in our laboratory. The practically we are not using any kit for isolating the DNA. We are using this particular method, and I will be sharing with you. And before coming into that, we need some formulas or some rules and regulations to be followed for doing the biomedical research. So what is it? So like for example, if nowadays everybody is being vaccinated, so if I want to study what is the effect of the vaccine on different people, whether it is being able to invoke the antibody response or whether it is being able to produce the T cell response, or otherwise at least the people are responding to vaccines or not. If we want to study that itself, we need permission to that kind of do such kind of research. I cannot just straight away go to some person and collect a blood sample and work on it. We have to follow certain rules and regulations. That is what is the called as the ethical guidelines, which are actually governing the biomedical research. So when a biomedical research is being done, this ethical guidelines should be followed. And this is being awarded by the ethics committee. It will be awarded by the ethics committee after undergoing or after having the discussions. Okay. And this is actually used to protect the dignity, rights, and well-being of the potential research participants. So anyone can come out of it at any time if they are not feeling comfortable, or they have their own choice of participating. Even though if a college is being interested to do a particular study, if a student is not interested, that student can opt out. So that is what it is been here. So Indian Council for Medical Research is giving certain guidelines to do this research, and this may be useful for evaluation of new drugs, for testing of some devices, some diagnostics, for manufacturing some vaccines, for preparing some herbal remedies, for doing some epidemiological research, for doing some human genetics research or transplantation research, to study the reproductive technologies, what has been available, and for many things it has been available. Okay, so after discussing our problem, we should not reinvent wheel. So the problem will be presented in front of the elite group, and they will have the discussions whether it is essential and how that can be modified and what number of samples should be taken. Like that, every discussion will go on, and further the permission for doing the research is being given. And when this permission is being given, we need to, after that we have to get the consent, informed consent. So all these days, everything goes on if we are being providing our willingness, I said. So that is being actually documented. It will be in the form of a sheet, and every detail of the work will be written there, why and what for it is being done, by whom it is being done, every detail is being given. And further, you have the participants will be, if they are willing, they will just sign, yes, I am willing to participate in this research. So that should provide exactly why that particular study will be given. And anyone has the refusal to opt out of these studies. And before initiating, when you are getting your consent is being obtained, you should have a questionnaire. What is meant by a questionnaire? So if there may be different objectives for studying, one of us may be interested, like yeah, fish is a good nutritional source that protects us from, yeah, a certain set of disease. Or you may tell red meat is something harmful you want to study. Or it is what is making people more prone to diabetic these days, whether it is because of lifestyle changes or because of the food what we are having, is it being contaminated or not that yeah, not that pure as like the olden days, like that. Are we not taking healthy diet? Many factors we can study. And similarly, if there is a disease occurrence and prevalence of any infectious disease also, the clinical spectrum of the disease may be varying. What is meant by that? Even though the people are being affected by that disease, there will be grades of the symptoms that is being shown by the individuals. And these gradations you can actually make out these changes and group them into different categories. For example, if we want to study the HIV infected individuals, this HIV infected individuals, they sometimes progress to AIDS stage, or sometimes not. And what makes them actually susceptible is there may be different causes. And as an immunologist, I may argue it is maybe contributed by the immune system. If a nutritionalist is studying he or she may be arguing the nutrition is essential and that is actually playing the role. And if at all someone studying the positive psychology is studying that, they may tell the positive psychology of the affected individual is making them live long. For example, when we did some studies with the HIV affected individuals, we could see that the women were able to sustain their CD4 count for a longer time. And when it was being analyzed, there can be different reasons. And one reason is like when they are having a family to take care, they have a hold upon something. When we have a hold upon something, we want to live and we want to do something to the children, then that makes even dragging the life also. So that may be the reason that women are being, and also we know that women are having better willpower and they can manage stress. That may be the another reason. One another reason is women are being diagnosed at an early age of HIV infection than men. What we are trying to say is in India, like whenever a woman is being conceived, when she goes for the pregnancy testing itself, she is being tested for the presence of HIV infection or not. When that is being done at a very young age, she is being diagnosed. But from through her only, the partner is being tested whether he is having the infection. When he is being tested, it might be a longer time. We don't know when exactly the partner got infected. That is the one reason. One reason maybe the women are able to maintain the CD4 count for a longer time and they have higher CD4 counts. And as well as the female hormones are playing a better role and helpful in production of more amount of the antibodies. And so that may be also giving a disease resistance mechanism for the female. And so for this, to collect all these information, we should have a questionnaire. That questionnaire will describe about the person's the disease, about the person's nativity, about the geography, everything, like it will be written, and about the disease also everything will be marked, and then you go on, go ahead. That is called the questionnaire. So when a questionnaire is being formulated, it should be very simple and logical questions should be only asked. And nowadays, it should be, you should be able to just tick A, B, C like that. You should give all possible answers and you should make them just tick. Okay, and that will be helpful. Okay, so now for doing the biomedical research, we need ethical clearance. Then we should get an informed consent. We should have a questionnaire, and then only we should go for collection of human peripheral blood. So blood is the one of the most common specimens collected from humans for DNA isolation. Why? Because the DNA quality will be good, as well as the quantity what we get will be also good. And there are different other sources also you can opt for. You can even collect from your hair. You can isolate from your hair. You can isolate from nails. Isolate from the buccal mouthwash. That is, you have squamous epithelial cells in the cheek. So when drinking water is being given and the person is asked to swirl it out and spit it out, then you can collect the water, and that water will be also containing the squamous epithelial cells, and from that also you can isolate the DNA. So there are various non-invasive methods also for the collection. However, the quality and quantity of the DNA what we get will be very less. So those DNA isolation methods will be applied in critical situations, like like when a person has been for example, the ancient DNAs, when it is being buried, or when there is people are reluctant to give blood samples, like that, at that those times you can do opt for that kind of methods. But when people are willing to give 5 ml of blood sample or 2 ml of blood sample, it is well and good. You can go for this kind of methods for collection of samples. So before proceeding to blood collect from the volunteer, the volunteer preparation is very essential, and the different needle sizes should be decided, and then tornics are essential. That is just you might have seen a strip kind of thing that is being tied in the upper arm before collecting the sample, like that. Then you should have the gloves. You should have isopropyl alcohol band-aids. If there is any bleeding, the band-aids can be put off. And then you should have a biohazard waste container. So biohazard waste container is very essential. And these biohazard waste containers will contain sodium hypochlorite, and that sodium hypochlorite will be essential for destroying the pathogens or whatever it may be, the microbes that is being present in the waste, biohazard waste. Then you should have tube racks as well as cotton, all those things you should have. Then you should prepare your needle, get ready the thing. Let me point out one more very important point to be considered here. Even though you are trained to collect blood sample from any individual, not everyone is actually ethically allowed to collect samples. Only those who are having a phlebotomy eligibility certificate, or that is called as phlebotomist, that person can only collect the sample. So that degree should be awarded from a medical institution, and it may be even a diploma course, but they should be a medical university should award the degree, and those, when they are having that certificate, then they are eligible to collect the sample. So this phlebotomist will be collecting the sample. Then volunteer preparation. You should not go and straight away collect the sample from the volunteer. What you should tell is, you should make the person comfortable, sit, ask the permission, explain them make the situation cool and calm, and then only you should collect the sample. So in these procedures for collecting the blood sample and all the procedures what will be explained, we have make it into small videos and that will be played for you. And yeah, but you can imagine now. You get a donor or a volunteer for giving the blood sample. You make them to sit calm, relax, speak with them. You can fill up the consent form. You can fill the questionnaire, and then you can ask them to fold their the left hand. You can collect the blood sample. You can make them to rest it on a table, and fold the thumb finger and then followed by the other fingers, and then just up and down the hand will be brought up and down. So we are going to collect the blood sample from the cubital vein. Cubital vein is the vein that you will see where your arm, yeah, you just look into your hands, you will see the vein. When you are doing up and down for 3 times, and then you just look into your hand, there will be a vein that will be protruding, and where they are going to collect the sample. And generally that is the place where for adults it is being collected, the sample is being collected. That is a easy place. So they at that particular place, the volunteer with the spirit, wetted spirit, the area should be wiped and clean, and then the sample should be collected. The sample should be collected, and there can be other places also, but for research purpose, those regions are not collected. On medical emergencies, those regions will be also sometimes used. Then from the vein, the sample is going to be collected, and now you have to transfer the blood sample, collected sample, to a tube. So where will we are going to transfer the sample. So where we are going to transfer the sample can have EDTA or without EDTA. So EDTA is what is EDTA? It is like anticoagulant, can act as an anticoagulant like heparin. For most of the experiments, molecular biology experiments, the sample will be collected in a tube that will be coated with the EDTA. So when the tube is being coated with EDTA, you are transferring the blood sample. When the sample is being transferred, what happens is like the blood will not get clotted. Otherwise, just when the blood is being taken out from from our system, free flowing nature, when it is being brought out, it will be just getting clotted, and this will be should be prevented for the DNA isolation. And so you are transferring it to a EDTA tube containing the EDTA. And after, how you are going to transfer it, you are going to remove the needle, and then you are going to use the plunger to release the sample slowly through the sides of the tubes into the EDTA coated tube. And afterwards you have to cap it out properly, and you will be mixing it properly. So all these procedures should be done after wearing the personal protective equipments. What are personal protective equipments? And what we are seeing in the COVID ward, the doctors are wearing, the nurses are wearing, that is the personal protective equipments. A long coat below the knee level. Then you should have gloves. Then there should be a mask. There should be a headgear. There should be a goggles to protect the eye. So all these things should be weared, and then the sample need to be collected, and it should be processed. Earlier days, we thought we were actually supposed to collect samples wearing the personal protective equipments, that was people were having the feeling that it is to protect the experiment mostly. So the experiments will be more sensitive, and so to protect the experiments, this was being followed. But now, in addition to that, you should also protect yourself from the infectious agents what is seen in the biological sample, and for that reason also these precautions need to be taken, and it should be collected, and it should be done. Okay, now after collecting when it should be mixed properly, and then the sample should be transported through ice to the laboratory for further processing. Okay, now let us come to extraction of genetic material from the human blood. So for most of the molecular biology techniques, they require the genomic DNA as a startup material for manipulation. And DNA extraction is the isolation and purification of DNA from various biological sources, including human blood, then cells, tissues, etc. You can do. And there are several methods that are actually employed for DNA extraction from the blood. But mostly the method depends upon how much quantity you need, okay, is required, because all the high throughput techniques relies upon the DNA quality. Care should be ensured to remove all the PCR contaminants and the inhibitors. Okay, for the example, the hemoglobin is a PCR inhibitor. So when you are doing the polymerase chain reaction, it can actually inhibit it. It can inhibit. So there is a well said saying that garbage in and garbage out in PCR. Whatever you give, that's a mission, right? So that is going to follow your orders. So so your input material should be also good. If you are giving a input material, proper input material, you are going to get a proper output material. Okay. So the steps of blood DNA extraction procedure, you have to collect the 5 ml of blood, and with the EDTA coated pack container, and you can process it. There are various methods of processing, and what we are going to discuss for today, and the video what you are going to see for today is about sodium acetate method. And this involves different principles that, like you have this experiment is being done as in a 2-day step process. And the first day step process, you are going to lyse the RBCs, the red blood cells. As you know, there are different components of the blood. Can I stop here and ask a UG participant to tell me what will be the primary constituents of the blood? Any UG participant can answer me. What will be the... Yes.
Erythrocytes,
Okay, good.
platelets,
Okay.
lymphocytes.
Okay.
And ma'am, part of lymphocytes like neutrophil, basophil, eosinophils, or
Okay. Good. You were very close to the answer. However, you can, the whole composition is called as the white blood cells. And then only you have the lymphocytes. Lymphocytes, neutrophils, eosinophils, erythrophils, basophils. Okay. Then lymphocyte, if you want to classify, the lymphocytes can be the T and the B cells. Okay. Otherwise, your answer was very correct, and the major constituents will be the platelets. You will be having the RBCs. You will be having the WBCs. Now, let me talk about the WBCs. Okay, so the WBCs they will be having, and you will be you will have a serum. Okay, the serum or plasma. If you are allowing the blood to clot, and then you are going to separate the liquid component, it will be called as the serum. And otherwise, the fluid component what generally flows in, that will be called as the plasma. And here let another participant answer. we have seen discussed different constituents of the blood, and among this, which will yield or which will be responsible or holding the DNA, genetic material. Let another participant, UG participant, can answer me, like what will be the, which should be our target to tap out the nucleic acid.
WBCs.
Yes, WBCs are white blood corpuscles. Okay, thank you. And that is the reason we have it as a 2-day step. In the first day, the major components or the RBC should be lysed and it should be completely removed, and whatever contaminants, proteins associated with that, everything should be removed. That will be the first day step. And the second day step, by the end of first day, we will be pelleting out the WBCs, white blood corpuscles, and that should be lysed, and next day the DNA should be removed out. And so that is, so we have, it is a 2-day step process. Okay, 2-day step process after a whole night incubation of the WBCs, white blood corpuscles, in the buffer. We will be next day we will be processing it for DNA. And so the some steps, critical steps will be the cell lysis. You should remove the lipids, and then you should precipitate the proteins, and then you should precipitate the DNA, and then you have to spool out, take it out, the DNA, and remove the salt residues, and then your pure DNA will be isolated. So this has been explained in this particular flow chart, and that is also explained in the small videos what my students had made it for you. And so the day 1 procedure will be, you need to transfer it to the blood sample collected to a 50 ml centrifuge tube, to a bigger tube you should put, and then you should add the RCLB, or red cell lysis buffer. So this preparation of this buffers, as well as the importance of reagent preparation, that also we have made it as small videos for you. That is the reagent preparation. We will have one video. So the reagent preparation is very important, and so you have to prepare a stock solutions. You have to prepare. Then you can improve the shelf life of the reagents. You can prepare stock and you can store it for a longer time, and then you can make working dilutions from that, and you can take little quantities of it and you can work it out. So directly, if you are going for working solutions, sometimes there will be chances of error about the molarity and normality of the reagents what we are preparing. So you should have the these reagents prepared at stock solutions, and then you can use it out. So these blood sample what you have taken should be washed. So here washing, what is generally washing? You will be adding water to something, and then you will try to remove the dirt. Here the washing sense, you are adding some buffers, you are mixing them, and you are removing out the unwanted material. That is the thing what is it is being done. And so you have you are when for washing we need a bigger material or tubs or something like that. So for that after transferring to 50 ml centrifuge tube, you have to add RCLB, red cell lysis buffer, in the ratio of 1:6. If there is 1 ml of blood, then you have to add 6 ml of the buffer, red cell lysis buffer should be added. Then it should be mixed thoroughly, mixed vigorously. Then you have to centrifuge, and you have to, when upon you are mixing with this buffer, it will the name itself implies that it is a lysis buffer of what? red cells. So it will be this red cell lysis buffer will lyse the RCLB, RBCs, and then so after that you will be removing the supernatant. Now you will be having pellet. This pellet should be once again added with the RCLB, then mix it, centrifuge, and remove out. Why you are going for centrifugation? Why don't you leave it like that in the stand itself for precipitation? No, because centrifugation is nothing but increasing the gravitational force in an artificial way, at a quicker way, at a faster way you are going to do that. So when you are being done with that, so your components get, some of them will be getting precipitated and some will be seen in the supernatant. So supernatant you are discarding, and once again you are adding if at all, until your what color your blood will be of red in color, and afterwards you are adding the buffer. And yes, ma'am. And when you are adding all these things, and when it is being impurities are red blood cells are being removed, the white blood cells have a whitish straw colored nature, and so that should come as a pellet. For that you need to wait, and to this you should add 50 mule of 10% SDS. So this was going to break down the WBCs, and this WBCs will be broken down, and now the DNA, which is the genetic material which is seen inside, should come out. So you are incubating this at overnight for 42 degrees Celsius, and you will be incubating, and then next day you are going for second day step. For the second day step, you are going to add 1.5 ml of 3 molar sodium acetate. So here you will be seeing different molarity of the reagents will be added. So here sodium acetate at a higher concentration you are using. So this is also the popularly called as the sodium acetate method or salting out method. And so the 3 molar sodium acetate will be added, and then it will be vortexed, and once again it will be spin and brought down. Then you have to transfer the supernatant to 15 ml tube, and then you should add ice cold isopropanol. Ice cold isopropanol helps in precipitating the DNA. Now initially the RBCs were there, that was actually removed, and then the protein was being removed from the cell membrane, everything what we got. Now you have the DNA, and that DNA, when you are adding the ice cold isopropanol, it will be getting precipitated, and that you can take it out. You can pause the video and you can watch step by step how it is being done, and then it will be spooled out, and this spooled out DNA will be taken in 70% ethanol for washing, and for salt, excess salt, anything should be removed, and for that you are washing it in 70% ethanol, and then you have to dissolve it in buffer, dissolve it in the TE buffer 200 ml, and afterwards the next day you can quantify it. So the quantification is being also explained as a small video, and this, because of this quantification, what happens is you can find out how much is your DNA concentration, how much is your RNA concentration, and how much is your protein impurities there. Like that you can find it out. Okay, and there are different methods of choice for DNA extraction, and the method what you are going to choose should give maximize the DNA recovery. It should remove the inhibitors, it should remove or inhibit nucleases, and it should maximize the quality of the DNA. So that is all essential. Okay, so whenever these tests are being done, safety procedures should be followed. So after collecting the sample, you have now removed the needle out, and so you should not. There will be a cap for this needle. You should not try to recap this and safe it properly. You shouldn't do that. Instead, you should go to a needle cutter. It is a small device, very small device, and you can show the needle, and the tip of the needle will get burnt up. So then for why this is being done is it is to prevent the needle stick injuries, needle prick injuries, if anything should not happen to the person who is collecting the sample. For that, all these things we are doing. Then you should dispose all the waste materials into the biohazard container, notified container. You may be knowing the biohazard symbol. The container should be kept separately, and there it should be transferred. Then always you should wear the gloves, mask, full length lab coat, headgear. For DNA extraction, it is better. Then you can work in a, you have to clean the working area with disinfectant, and then you need to work. Okay. With this, let me finish my presentation, and can the organizers please share or stream the video?
Ma'am, we will post the, ma'am, we will upload the videos in the Google Classroom.
Okay. They are not going to watch it now?
No, ma'am.
Okay. If they are going to watch, is there a possibility, sister?
That is, I will share now.
Okay, you can just check. If it is possible, then they can get
Yes, ma'am.
clarifications. It will be easy.
One minute, ma'am. I will. Ma'am, Jayalakshmi ma'am. Jayalakshmi ma'am.
Yes, ma'am.
I shared the videos, but there is no sound. There is no sound when I shared in the Zoom platform.
Okay.
So I will upload in the Google Classroom, sir.
Yeah, you can upload, but just let it just move without voice. Also, let it move in your screen so that yeah, they will recollect what I have spoken to them.
Okay, ma'am. Okay, ma'am. வருதா இப்போ சவுண்ட் வரலன்னா வருதா அந்த இது வருதா இமேஜ்
You have to submit in your office, I think.
Already done.
Ma'am, ma'am, Jayalakshmi ma'am.
Yes, ma'am.
Ma'am, I have shared the first two videos.
Okay.
The reagents, preparation of reagents, and then the how to take the sample, blood sample.
Yeah, there are 2 more videos for
Two
today.
more videos are there, but without voice, the understanding of concept, it's very difficult, ma'am.
Already it is explained, no, ma'am.
Shall I share?
Yeah, are the participants in a hurry to leave the meeting, or do you have any time constraints? If you have time constraint, you can close the session, or if not, you can share it.
They are asking feedback link, ma'am. Shall we ask in the participants?
Yes.
Participants, do you want to share the videos in the Zoom platform? Otherwise, we will upload the videos in the Google Classroom. If you want, yes.
Okay, thank you, ma'am.
Okay, ma'am. Kindly wait 1 more minute, ma'am.
Okay.
Participants, the forum is open for discussion. You can ask your queries. Participants, otherwise you can post your queries in the chat box.
Okay, we will be posting in Google Classroom. No worries with that. So you can have your clarifications.
Participants, we will upload the videos in the Google Classroom. Don't type the message again and again. Okay, you can ask your clarification or you can ask your queries. Ma'am, there is no query, ma'am. We will move on to the formal vote of thanks. We will meet tomorrow. Okay. Lakshmi?
Okay, thank you.
Ma'am, one question is posted. One question is posted in the chat box. What to do if needles used contaminated due to any reason and the person gets affected?
Okay, we should see nowadays, these days, only we are using disposable needles and syringes, and it is not, it is not, what is it? Reused. Needles are not being reused. But if it is been, somebody got injured, then they should immediately report to their higher authorities. That maybe if you are a research student, report to your supervisor. If you are a practical, doing some practical, you can report to your staff in charge. If you are working in a laboratory, you can report to the doctor who is present there. And then the patient or from whom we have collected the sample, that those persons should be studied for any ailment they are suffering from or not, any infectious disease if they are having or not. If at all the person is being affected by some of those kind of diseases, what you can do, if there are some antibody therapies available against that infectious disease, the person can take antibody therapy, because if antigen is being given to a particular person, then there will be memory cell response being developed, and so there will be immune response being developed. And before allowing that itself, this, when you are giving artificially prepared antibodies, and then that can go and block the pathogen which has been got injected, and it can neutralize the pathogen and it can destroy it. So that will be the explanation for that. And there are some kind of situations where they when they were collecting sample from a HIV person, it got accidental struck, and at that time, then they should actually report if the person is being getting affected because of this. any kind of insurance that could be claimed because of this occupational hazards, then that can be claimed. Okay, so that is the another thing. And what is the importance of questionnaire of used in research? So these questionnaires are nothing but a set of questions framed to support your hypothesis, details that you will be collecting for supporting your hypothesis. That should be that is what. So any other question? Any other doubts? Okay, thank you, ma'am.
Lakshmi.
Thank you. Thank you, ma'am, for enriching us through your wonderful presentation. Now I invite Dr. Hriday Antoinette Sophia, Assistant Professor of Chemistry, to propose the vote of thanks.
Gratitude is the most exquisite form of courtesy. I deem it a great pleasure to propose the vote of thanks on behalf of the organizing team. First of all, I would like to thank the Lord Almighty for his gracious blessings and abundant guidance bestowed on us. My heartfelt thanks to our resource person, Dr. M. Jayalakshmi, for sparing her precious time and enriching the young minds with her lucid lecture. Your session was very interesting, ma'am. Thank you so much. I'm sure that the students would have gained still more knowledge about the genomic DNA extraction. You have clearly explained the stepwise method for extraction, the safety procedures that has to be adopted, the reagent preparation, and the blood sample collection. Thank you, ma'am, once again. My special thanks to our Secretary, Reverend Sister Flora Mary, our Principal, Dr. Sister A.S.J. Lucia Rose, and our Deputy Principal, Dr. Sister Kolandai Teres, for their constant support and encouragement. I take this opportunity to thank Dr. Sister Araki Genesius Alphonse, overall Star Coordinator and Member Secretary, Star College Scheme, for her committed effort and dedication. I owe my gratitude to all the heads of various departments, members of the organizing team for their untiring efforts. Special thanks to the technical team for their uninterrupted continuous support. Last but not the least, I thank all the participants for your active participation. Thank you, one and all.
Thank you. Thank you. Thank you all.
Thank you, ma'am.
Thanks.
Dear participants, feedback link is posted in the chat box. Kindly fill it. An important announcement, dear participants, kindly note, you will have virtual training, hands-on training session from 3 to 4. Videos will be posted in the Google Classroom. Go through it properly. This will be asked based on both the virtual hands-on training as well as on the session. So kindly go through it properly. You will be having virtual hands-on training session from 3 to 4. Videos will be posted in the Google Classroom. You can have to go through it properly. Quiz will be based both on the session as well as on the hands-on training. Okay, another announcement, participants. Tomorrow also we'll be having the session from 10:30 a.m. Feedback link is posted in the chat box. There will be virtual hands-on training from 3 to 4 p.m. Tomorrow the session will be from 10:30 a.m. Thank you, dear participants. Thank you, ma'am.
Yes, I am signing out. Thank you. Meet you tomorrow.
Okay, ma'am.